Hey, what's going on, biologists? Today's topic is going to be the cell. It's going to be fairly straightforward, unlike the chemistry stuff that we've been doing up until now. I actually like the cell, and hopefully you will too. And I'll turn my camera off so that you can concentrate on the PowerPoint presentation. Here we go. Okay, everybody, if you are alive, then you're made up of cells. It is true. Everything living on the planet is made up of some sort of a cell. And anybody want to take a guess as to what you looked like 9 months before you were born? That's right. Every one of you looked something like this. Every one of you is derived from a single cell that divided many trillions of times to eventually make you. But not all organisms are multicellular. There's a lot of other cell types out there. Many of them, in fact, the majority of them are actually single-celled. And these are just some examples. Here's a single-celled amoeba. And these guys are very prolific. There are many thousands, if not tens or hundreds of thousands of undescribed single-celled organisms. Of course, the organisms that we identify with the most are actually things like this. This is a sequoia tree, and then, of course, this is a humpback whale. These are really large organisms in complete contrast to that very small organism we have in the upper right there. So, what is the biggest cell in the world? Well, technically, an egg is a cell. And so if you take that to its ultimate conclusion, the largest bird in the world, that would be the Voron patra, has the largest shell in the world and therefore the largest cell in the world. Of course, these guys are extinct. They don't exist anymore. But they did stand about 10 to 11 feet tall and they weighed about 1,000 pounds. And this little gentleman here is holding on to the remnants of one of these Voron patra eggs. That's a humongous egg. Now, this is all of it kind of in a nutshell. What we're going to learn about are the basics of all of the intricate organelles, the little components within the cell, and all of their functions, all the things that they do. There is a nice little Discovery Channel video that I have already posted to the Blackboard website. I urge you to check it out. It's only 3.5 minutes long, but it does kind of spell out all the major parts of the cell, what cells are like. It contrasts different kinds of cells, so definitely go and check that out. We are now going to move on, so if you want to pause here and go and watch that before you come back to the rest of it, that's great. This is a perfect spot. But I'm going to go ahead and move on to the cell theory. What is the cell theory? Well, it's really just this idea that every organism on the planet, everything that is alive, is composed of either one cell or more than one cell. And the cell really is the fundamental unit of life. That is to say, if you get smaller than the cell, you are no longer dealing with living things. So the cell is the fundamental unit of life. And also, the continuity of life from growth and division of a single cell is also a feature of the cell theory. That is to say that all of the cells that make you up right now, all the trillions of cells that make you up, originally came from other cells and that came from other cells before them until you go all the way back until the point of conception where you essentially derive from a single cell. So that is true of all living things, and in fact that one cell that you are derived from came from a sperm and egg cell from your parents. Okay, and that is the continuation of their cellular lineage as well. So you are the product of a cellular lineage that goes back many hundreds of millions of years. So you should be very proud of yourself in that regard. Here's a cell, and it answers the question, what is a cell? Well, this is a cell right here. Take a look at this cell. The definition that we're going to function on is this one right here. A cell is a tiny mass of water, proteins, and other molecules surrounded by a thin membrane. Well, that's pretty interesting. It's just a bag of stuff, essentially, floating in water. Of course, we know it to be a whole lot more detailed than that. What does a cell actually do? Well, it doesn't matter if you are a single cell or a multicellular organism. This is what the cell needs to do in order to actually be alive. They need to be able to acquire energy to survive. They respire, and that word will mean more to you after next week. And in order to respire, cells need oxygen, they need water, and all cells that respire and do metabolism produce waste products. And carbon dioxide is the classic waste product. Cells tend to reproduce, that is, they make more cells. They also respond and react to the environment around them. They synthesize materials for growth and maintenance. And in certain organisms like you and me, we have specialized cells that synthesize specialized materials for maintaining our bodies. Now, the point I want to make here also is this list of things that all cells do. I have a picture of a lion up there, as in this lion has to be able to do all that right there. It's cells. But this pertains also to single-celled organisms too. Single-celled organisms must do all the exact same things. Okay? And that's because this multicellular organism up here, this lion, is made up of many different kinds of cells. Does that make sense? And since each individual cell has to do this collectively as an organism, this also defines the organism. I want you to keep this in mind when we go through this entire lecture. It doesn't matter whether you're a big cell or a little cell, you are going to rely on all of these processes here. Now, here's another feature of cells. Despite the fact that that egg we saw just a few slides back was enormous, most cells are small. And it doesn't matter here whether you're this little mouse or whether you're this elephant. This elephant may have more cells, but the cells themselves that make up the elephant are no bigger than the cells that make up this mouse. Well, that's interesting. So large organisms and small organisms are made up of cells that tend to be about the same size. That is, they tend to have about the same surface to volume ratio. Now, this is a very difficult concept to understand, surface to volume ratio. And I have a couple of flash animations on Blackboard for you to play around with surface to volume ratio. Now, bigger cells tend to have less surface area relative to their volume. Now that's kind of important. We're not talking about the size, we're not talking about the volume, we're talking about the ratio of the size to the volume. Does that make sense? So in this case, if you were to take the surface area of something, like this elephant right here, we're going to take the surface area. The surface area is a measure of a squared function. Think of it this way. If you were to measure how much carpet you needed to put in your house, what you would do is you'd take a ruler, you'd measure the width of your room versus the length of the room, and that is two dimensions. That's the width times the length. That's a squared function. So you would need like 200 square feet of fabric or 200 square feet of carpet. It would be something measured in squares. However, if you were trying to measure the volume of the room, let's say you wanted to fill the room up with water. I don't know why, but maybe you want to do that. Then you need to take the length times the width of the floor times the height of the room. So length times width times height. That's 3 dimensions. That would be a cubic measurement. So instead of saying square feet, which would just be the floor, you would say cubic feet. Cubic feet is a measure of volume. So when we're talking about organisms getting bigger or smaller, their surface area changes on a square, but their volume changes on a cube. In other words, they don't change together. Okay? This also means for cells that the bigger you get, your surface area does not increase as fast as your volume does. And it's possible to actually get to a size where you cannot move materials into and out of the cells efficiently enough to get much needed gases or sugars or food to the center of the cell. In order to do this most efficiently, it actually helps for cells to remain small. Now, I want you guys to be able to do this little exercise here. We're going to do a little math exercise. Not a huge amount of math, okay? And notice that on these cubes that I'm showing you here, I don't actually have units. I only have numbers. But these can be whatever units you want. You can call them inches, you can call them centimeters, you can call them meters, whatever you want to do. It's fine. But I want to make a point here. I have here SAs. SA stands for surface area. V stands for volume. SA over V is the surface area to volume ratio. Okay, so let's go ahead and calculate these three things for these 4 cubes. Are you going to have to do this on an exam? Yes, yes you are. So, slide number 10, I strongly urge you to take a look at this slide a number of times, maybe replay this portion of the video a few times, and maybe even get out a blank piece of paper and write this stuff down yourself. Try it out yourself. Okay, let's start with the easy one, this small cube down here. This small cube, how do you calculate the surface area of a cube? Well, this is just standard math here. We're going to calculate the surface area of one side, and then how many sides does a cube have? It has 6 sides. So whatever surface area we have of one side, we then just multiply it times 6. So surface area of this cube is 1 times 1, which is 1, times 6 sides. So the surface area of this cube is 6. Now, this guy right here, the surface area would be 2 times 2, which is 4, and then 6 sides, right? So 4 times 6, which is 24. The surface area here is 3 times 3, which is 9, and 9 times 6 sides is 54. And then 4 times 4 is 16 times 6 sides. If you do that math, it ends up being 96. 96 right here. So if you've written all of these down, we should have 6, 24, 54, and 96. Okay. Now, volume is calculated differently. Remember, volume is the length times the width times the height. All 3 dimensions multiplied times each other. That's the volume. In the case of this cube right here, it would be 1 times 1 times 1, which would give you a 1 right here. This number right here would be 2 times 2 times 2. 2 times 2 is 4. 4 times 2 is 8. So 8 would go here. This one here would be 3 times 3 times 3. 3 times 3 is 9. 9 times 3 is 27. And this last one here is 4 times 4 times 4. 4 times 4 is 16. 16 times 4 is 64. Now, you will notice, I'm going to go ahead and flash these numbers up here. We've done these first 2 rows here, right? You'll notice that as I go from a small cube to a large cube, my surface area grows, doesn't it? And so does my volume. My volume also grows. But remember, they're not growing at the same rate because one is a measure of squares, the other is a measure of cubes. So what I want to do now is take the surface area number up here divided by the volume, and if I start over here, surface area divided by volume produces a surface area to volume ratio of 6, or if you want, you can say 6 to 1. This is 24 to 8, which is the same as 3 to 1. Okay, 24 divided by 8. 3 to 1. This one is 54 divided by 27, which is 2 to 1. This is 96 divided by 64, which is 1.5 to 1. So again, as surface area, as the cube increases in size, its surface area also increases, as does its volume. But the ratio of these two actually shrinks as you get larger. So the question is, or you could actually do it the other way around. Start really large and get small, and what happens? Your surface area relative to your volume actually increases. Which means that cells, the smaller they get, the more surface area they have relative to the amount of volume they have. This is precisely why cells are small. This guy right over here is the ideal situation. Now, you probably already know this to be true, at least intrinsically. Let me give you a great example of that. Here's a bunch of students that I took out on a field trip with me, and it was a nice day out, but it got kind of chilly, wind was blowing. And this person right here got really cold after only 20 minutes of being out on the boat. Meanwhile, this guy over here, look at that. He's got the cut-off t-shirt. He's got the redneck t-shirt going on here. He's got the shorts going on, no shoes. He's not cold at all. Why is he not cold? Well, you might say because he has that extra insulation going on there, right? He looks a little bit overweight. But you know what? Actually, it has to do with the fact that as you get bigger, you actually have a bigger frame here. His volume may have increased, but relative to his surface area, he actually has less surface area relative to his volume because he's a bigger dude. Meanwhile, this poor young lady over here, as she gets smaller, the amount of surface area she has in contact with the air relative to her volume actually goes up, so she gets colder faster. You can try this experiment with your little 8-year-old cousin. Next time you go to the lake or the ocean where it's 55 degrees in the water, throw them in the water along with yourself and see which of the two of you gets cold faster. Chances are they're going to get colder faster because they're probably smaller than you. Another, and so here, this guy right here, yes, his volume is large, yes, his surface area is large, but relative to each other, his surface area to volume ratio, he actually has less surface area compared to his volume, compared to her. She has a greater amount of surface area relative to her volume. Here's just another demonstration of that. In this case, they're illustrating here with a small cube versus a large cube. And if you wanted to take this large cube and actually get the same surface area to volume ratio out of it as this small cube, all you got to do is chop it up. Look at that. If you chop it up, all of a sudden, your surface area to volume goes up. Isn't that amazing? How about that? So you can achieve the same surface area to volume ratio as this little guy here in this amount of size just by chopping it up. That's kind of nice. Okay, so surface area to volume ratio. Yes, there will be questions on this on midterm number 1. If you are having trouble with it because it is a little difficult concept to understand, definitely send me an email and let me know. Now, let's go over the different kinds of cells. There's really 2 main kinds of cells, 2 major varieties, and I'm showing you them here. And we're going to go over each one individually, but here are some basic features of all cells, right? Basic features of all cells, you have to have a plasma membrane around your whole thing, okay? This is like your outer coat. It's your plasma membrane. You also have to have some genetic material here to carry your genetic information, your genes. Your chromosomes are the things that you have that carry all that information. You also need to have the means to make proteins, and ribosomes are the surface on which proteins are made. These 3 things are the requirements for all cells. Doesn't matter what kind you are. However, there are other things involved, very important. So these are the 2 cell types. Eukaryotes are the first one. Notice how large they are relative to this little dude right over here. This is a prokaryotic cell. Prokaryotes are much smaller than eukaryotes. If eukaryotic cells were school buses, then a prokaryote would probably be more like the size of a softball. Okay, so you can imagine a school bus with a gazillion softballs all over the surface. If I were to take this prokaryote and blow it up, this bottom picture is what it would look like, okay? But realize that this prokaryote is actually smaller than the eukaryote. Okay, so what other differences do we have between these two? They're actually profound differences, and we're going to talk about the eukaryote first. Eukaryotes are actually the more advanced of the 2 varieties. Eu, in scientific lingo, eu means true. Karyote refers to the nucleus or the genetic material of the cell. Eukaryotes have a true nucleus or a true separate compartment in which all of the genetic material kind of sits. So, distinctive characteristics of a eukaryote. They possess a nucleus, which is the packet of genetic material within a thin membrane. In this particular picture, it would be this central little green disk area. That's the nucleus. That's the place where the DNA resides. Okay? Now, the other characteristic of eukaryotes is organelles. Inside of this cell are a whole bunch of small little machines, little tools that are all compartmentalized within there, each one having its own individual separate task. It's kind of like having little separate Ziploc baggies in a lunch pail, and in each Ziploc baggie you have a different food item, and having them all separated out by Ziploc baggie means that the food items are not going to accidentally mix together so that your potato chips end up tasting like your orange juice, okay? Keeping things compartmentalized is a really good thing when you're talking biochemistry. Remember how we kind of talked about complex biochemistry? We're going to get more into that a little later, but we've already talked about some chemistry things. And if you start mixing chemical pathways together, they kind of interfere with each other and screw each other up. If you keep those biochemical pathways in their own separate compartments, they're less likely to interfere with each other. That is the advantage of eukaryotic cells. All these little organelles are separating out the different functions within the cell. That's kind of nice. Now, floating are the organelles within the cytoplasm. The cytoplasm is like an aqueous fluid, an aqueous medium. It's actually like a matrix within which all these organelles are kind of suspended. And we're going to talk a little bit more at length on each one of these. So eukaryotic cells actually are comprised of many different kinds of living organisms. Animals are one such example. So this is an example of an animal cell here, and we're going to talk about the difference between animals and plants in just a second. Another kind of eukaryotic cell would be plants. So animals and plants are actually both within this eukaryote category of advanced cell types. Plant organelles are kind of different here. The number one organelle that is different would be cell wall. Animal cells do not possess a cell wall. Animal cells are kind of squishy. Plant cells are rigid because of this cell wall. This cell wall is actually what makes bark and wood so hard. But it also is what makes carrots and broccoli have that stiffness to them where they're not kind of droopy. So actually it's very important. It gives structural support to plant cells. Number two is a central vacuole. In this picture here, you'll notice there's a huge balloon-looking structure, and the vacuole is kind of important for plants because it allows them to store certain materials, like for example starches, sugars, or even waste products they can store in there. And lastly is the chloroplast. Swimming around or floating about suspended within the plant cell, these chloroplasts are the center for photosynthesis. We're going to cover photosynthesis in an entire lecture unto itself in the future here. So these are the major types of eukaryotes. There are others, but we're really just going to deal with animals and plants for right now. And I do want you to remember these three organelles that differ in plants that are not present in animals. Now, let's jump really quickly here to the prokaryotic cells. These are that other much smaller variety of cell, and these are slightly different in that they do not possess a nucleus. That is, the genetic material that otherwise would be found in a nucleus. In a prokaryotic cell, it's found just kind of strewn about, floating in the matrix within a prokaryotic cell. So the cell also lacks many of the organelles, if not most of the organelles, that you might find in a eukaryotic cell. And this group includes all the bacteria and also one of their cousin groups or a sister group called the archaea. But all bacteria, you guys have heard of bacteria. Bacteria fall into the group of prokaryotes, and these are an ancient group of organisms. In fact, on the tree of life, you could kind of consider them to be kind of like this. At the very base of the tree going back probably about 3.8 billion years, that's 3.8 billion with a B, includes the bacteria. In this case, we're calling them the eubacteria. Eu again means true. So true bacteria, and then the archaea. These are the two the most ancient groups of life on this planet to date. This one lineage that kind of comes up here splits into four main groups. We've talked about animals and plants, but we don't want to forget that the fungi, which includes the mushrooms, molds, and slimes and fungus, that kind of thing, all of these things are also considered eukaryotic cells. But then there's also protists, single-celled advanced cell types, but single-celled organisms nonetheless. They float around in the ocean and in streams and rivers and lakes, and also many of them are terrestrial as well in soils, etc. But this group right here, we're really going to focus just on plants and animals for now. We will come back to these later on in the semester, and we also will revisit the archaea and the eubacteria as well. So as far as cell structure goes, all cells have this plasma membrane, okay? And they also have this DNA region and cytoplasm within which all of the organs and organelles are suspended, okay? Now, plasma membrane. Let's focus on some of the organelles at this point. Let's look specifically at the plasma membrane. This is a thin membrane that surrounds the entire cell, and it separates the internal environment from the external environment. It is what contains all of the goodness inside, or the nuclear goodness inside the cell, and it keeps it separate from the ocean, the lake, or whatever it is that the cell is immersed in. Okay? And now, at the site of the plasma membrane, it's almost like it is fitted with hundreds of doorways throughout the entire surface of the cell. And it is through these doorways that the exchange of materials is controlled. Some items are allowed in, others are excluded and kept out. This is also the site of gas exchange and diffusion of molecules into and out of the cells, and also active transport of materials into and out of the cells. Water is usually allowed to pass into and out of the cells unimpeded, along with a few other ions. But there are some notable things that are not allowed into the cell, or actually are kept inside the cell, that are excluded from being able to pass across the plasma membrane. And if you remember lab number 2 in the Biology 109 lab series, if you've taken that, we did this experiment with dialysis tubing. Okay. In the cell membrane, in that plasma membrane, is a whole series of proteins that are kind of set into there that serve as markers, and they either serve as a channel protein, which creates like a tunnel or a doorway, which only allows certain things in and out. Also a transport protein. So in some ways, instead of it creating a conduit through which something travels, something will actually latch onto it, causing it to flip and rotate the protein or whatever it is that it's trying to transport into or out of the cell. It acts almost like a doorman. You guys ever seen those circular doorways, those revolving doors? Well, imagine that in number 1 here, the channel protein. In that case, it's like it allows things to just go right through the revolving door. But if it was a transport protein, then the item would come up to the revolving door, and the doorman would have to grab it by the hand and actually escort it inside. That would be kind of the analogy there. There's also cell recognition proteins. So on the surface, you have these strange little proteins that are sticking out that bind to specific signals that are floating out inside the environment or inside of the body in which you find these cells. And these proteins, for example, would tell you what is yourself, what is actually you, versus what is a foreign cell, like an invading bacteria or something like that. The reason why your body can identify bacteria and other invaders is because they do not possess the same cell recognition proteins on the outside that your cells have. Receptor proteins are also really important. These bind to molecules that are passing by, and these molecules may signal to that cell. Let's say it's a hormone kind of cell, a cell that produces lots of hormones. By having a particular molecule come along and bind to it, it signals to that cell, hey, it's time to start putting out more hormone. maybe there's not enough hormone, and having a drop in the hormone levels inside the cell actually triggers this cell or I should say, triggers this binding molecule to come along and cause it to make more of that hormone. So these are some typical ones. There's a couple of other interesting ones. Enzymatic proteins. These are actually proteins that on the surface that carry out some sort of a metabolic process. Like I'm showing you these red blood cells. These are red blood cells in your blood. They are loaded with enzymatic proteins that help it take on oxygen and also take on carbon dioxide when they're removing it from your body. Also, junction proteins. That would be like proteins that connect one one plant cell to another plant cell next to it, so that the cells can actually communicate with each other by sending fluids or sending other proteins through these jointed locks. This slide right here kind of just illustrates a whole myriad of different proteins that hang out on the surface of a cell. Again, those that help allow things in, either by just providing a channel or by assisting it through, or in this case, these are proteins that actually are out there detecting what is floating around in the surface. So these are all kind of important functions of that cell surface. It's not just a barrier like a chain link fence keeping things in or out. It actually is far more complex than that. Okay, so that is the cell membrane, but there's a whole lot inside the cell. And each one of these organelles, we're going to cover them in the basics, and we will go over some of them in much greater detail in future lectures. So we're just going to do like a survey of all of the organelle types in the body right now, starting with, I should say, here's a difference. These are animal cells here. And this here is a plant cell example. And remember that there are a few organelles that are slightly different in plants than you would find in animals, but you do need to know all of them for the plants and the animals. And so let's look a little bit more detailed at that plasma membrane again. You'll notice that it looks almost like there are these red balls with these golden tails hanging down, and you'll notice that they're kind of, the ones here on the bottom are upside down. And that's because these are all made up of phospholipids. We had mentioned this in the biochemistry lecture last time, that the cell surface is actually composed of a whole series of phospholipids kind of lined up together. The polar heads of these phospholipids are all kind of attracted to each other, and the nonpolar tails are all attracted to each other. So just spontaneously they line up this way. Now, if you took this one sheet and you put it into the presence of another plasma sheet, they end up inverting so that the nonpolar tails are touching with the nonpolar tails of the other molecules next to them, and they make these sheets. Well, that's kind of nice because it means that you can produce a really interesting barrier, as we mentioned before. Water will come in and hit this, but it won't penetrate through automatically unless it has some sort of a channel or something that it can go through, because it's going to eventually encounter here in the center the nonpolar tails of this bilayer, of this what we know as a phospholipid bilayer. So this makes it pretty much impenetrable to most things. Now, things can still transfer through, but it's now controlled because you're fitting these proteins in to the plasma membrane to allow only certain things in and out. So that's kind of convenient. And let's not forget about the cytoplasm, right? The cytoplasm is that aqueous matrix fluid where molecules and organelles are all kind of suspended throughout it. And now we kind of think of it conceptually as a bag of stuff, but actually it's more complex than that. And this picture right here on the right actually shows you the girders and struts and joists, all of the supporting structure that has been stained so that you can actually see what the matrix looks like of the cytoplasm. It's actually quite impressive. And we call this the cytoskeleton. The cytoskeleton. It's not a skeleton like bones per se, but it does serve the same function as the bones in your body in that it maintains the shape of the shell. So you have here the shape of the shell. I mean, it's kind of like it maintains the shape of the cellular three-dimensional space. So these are made up of protein filaments and tubules that kind of all are interconnected and touch with the plasma membrane to hold it out so that it doesn't collapse in on itself. Now let's move to one particular organelle type. Where is the DNA stored in a cell? Well, the answer is, you probably know it to be the nucleus. Now, before I answer or go into greater detail on the nucleus, though, let's just revisit why it is that organelles are even maintained in their own little compartments, why it is that they have their own little membranes around themselves. You can think of it as an analogy this way. Why do you have skin around you, and why is it that all your organs are separate? Think about the biochemistry that is going on in your stomach. In your stomach, there's all kinds of acids and, really noxious stuff that you need to have in order to digest your food. Now think about the biochemistry that's going on in your heart or your blood. Obviously, that's different biochemistry, and you wouldn't want your gastric juices spilling out into your blood system, right? That would be a really bad thing. Fortunately, those organ systems remain separate so that the blood can do, its blood stuff really well, and the stomach can do its stomach stuff really well. The analogy holds really well because inside the cell, each little organelle is doing its own separate thing, and keeping them separate allows them to function even better. They become even more efficient. So compartmentalization is the key to this. It's the name of the game. It separates areas for specific functions, and it means that areas can be specialized for metabolism. You can have things that actually specialize in breaking things down, and other things that are specialized in building things up, and you don't have to worry about them interfering with each other. So back to that nucleus, the organelles. Let's talk about the nucleus as an organelle. It is a membrane-bound compartment, and it is where all of the genetic information, or the DNA, is stored. So when you want to access that information to build whatever the DNA instructions have, you need to go into the nucleus, get that information, bring it out of the nucleus, and then do your assembly outside. That is a process called protein synthesis, which we will also cover in a future lecture. Now, the nuclear membrane is porous, but it's only porous by a small amount. These are really, really small openings that only small strands of information can leave from. So large things cannot pass in and out of this. In the center of the nucleus is a dense area called chromatin. And it is like a concentration of DNA and protein kind of all wound together. And it stays there when the cell is just doing its everyday normal functions. When the cell gets ready to divide and make, two of itself, well, then the nucleolus actually does some interesting things. But for right now, this is kind of what it looks like. It's just this dense mass area inside of the nucleus. And the nice thing is that it is the site of ribosomal RNA synthesis. Ribosomal RNA is very important for the formation of proteins, a topic that we're going to discuss a little bit later. Okay, let's talk about ribosomes now. Ribosomes are little nodules or chromatic bodies that are outside of the nucleus, and it is where the proteins are built. And these are like basically factories waiting for instructions to make proteins. And where do the instructions come from? They come from the nucleus. That's right. So the DNA, the information gets out from the DNA in a process that we're going to cover later. And it's almost like a little photocopy of a section of DNA. And that photocopy comes along, and the ribosome actually reads that little photocopy of the DNA and spits out a protein. That's how that process works. Now, let me cover the basics here of protein synthesis. We're going to learn about the details of it in a separate lecture, as I mentioned before. The first step in this process is called the endomembrane system. And it is a series of folds and convolutions that come off of the nuclear membrane or the nuclear envelope. And so when the information from the DNA passes out of the nucleus, the first place it goes is to the endomembrane system. And the endomembrane system is actually made up of a number of parts, and we're going to talk about each part in sequence. And this gets kind of tedious, so if you have to rewind and watch this over, especially after reading your textbook, it will really, really help. So there are three main parts to the endomembrane system coming off of the nucleus. The first main part is called the endoplasmic reticulum. The endoplasmic reticulum does a couple of things, and then it passes the product on to another system called the Golgi bodies. The Golgi bodies do some more stuff to those proteins, and eventually they get transported out through the third part of this called the vesicles. Now, what is the analogy here? I love the analogy of a car assembly line for explaining the endoplasmic reticulum, the Golgi bodies, and the vesicles. So let's focus first on the endoplasmic reticulum. It is the membranous compartment that is actually attached to the nucleus. And notice that it's granulated here. I actually show the little red dots all over the surface on this part, but not on this part over here. And that's because the one that has these little granulations on it is known as the rough ER, or the rough endoplasmic reticulum. It's rough because it's got the ribosomes all over it. Remember we talked about the ribosomes as being the place where proteins are synthesized? So proteins are synthesized on the surface of the rough ER, where all the ribosomes are. After it gets processed here and basically pre-assembled, it then gets transported to the smooth ER, where further processing occurs. And so in animal cells, this is continuous with the nuclear membrane. A little aside is that it's not so with plant cells, but we're not going to worry about that. What I'm showing you here is the major assembly portion of producing a car. This particular car happens to be the Nissan Leaf, but we can refer to it as the all-new 2012 Nissan Protein, if you will. And obviously this car is not done, but all of the major parts have been put on and it's ready for further assembly. This would be kind of like what's happening in the ER. Okay, that's the main part. So remember, there are two regions here, the rough ER and the smooth ER, and these are the guys that are producing the lysosomal enzymes that actually help you digest materials inside the cell. We will cover that in a little bit. Let's first focus on the rough ER and then on the smooth ER. Remember, the rough ER is where the proteins are initially being put together. Okay, rough ER. These guys are arranged into flat sacs, just like this. And remember, what are those granulations? They are ribosomes. That's right. I know you said it. So that's what makes this the rough ER. The ribosomes on the surface give it that rough appearance. And the cells that are specialized for protein secretion, the ones where that's their whole purpose, they have a whole lot of rough ER. Like this picture right here, this is a specific cell in animal that produces a huge amount of protein secretion. So it is packed with rough ER. Now the smooth ER is the interconnected tubules that are on the outside of the rough ER. They're kind of continuous with them, but they have a slightly different purpose. They don't have ribosomes. So by the time they get to the smooth ER, the proteins have already been at least assembled at the most primary level. They haven't been finished yet, but they've been primarily put together. Now inside of the smooth ER, Lipids are usually added to the proteins to make them a more complex molecule. Usually the molecules floating around in your body that do things are more than just a protein. They have other stuff hanging off of them. And it is in the smooth ER that they are essentially put the finer touches on it. This is where the leather seats are put into it, and where the special stereo is put in, and the spare tire that goes in the trunk. That's kind of what is going on at the smooth ER. Now, kind of important that the smooth ER in your liver has a particular function of inactivating wastes like drugs or even alcohol. So when you take too many drugs, even legal prescription drugs, the more you take, the more you wear out your smooth ER's ability to inactivate them, and then you end up having to take more drugs to get the same effect. So the reason why it takes more drugs to get the same effect, you're wearing out your smooth ER in your liver. That's essentially what's going on. Now that second system after, well, I should say the rough ER, the smooth ER, the next system is the Golgi bodies. The Golgi bodies are the kind of like the finishing organelle. It is where the final touches get put on for delivery. Now it's kind of like, these cars here, these are all Nissan Leafs, the fully electric car. You know how you've seen them kind of going down the street and they wrap plastic wrap around them to protect them, and maybe they'll put some extra sealant on the outside, and maybe they'll box them up a little bit. They'll make them perfect for transportation. And that's kind of how this works. They essentially package the molecules for shipment to their final destination. That's the Golgi bodies. Okay? Now, here is a slide. This is slide number 38, showing you the full chain of events that goes on, moving from the nucleus all the way out to the product leaving the cell. Okay, so make sure you understand this order of events. We're going to go over the details of this again in a separate lecture. But number one, polypeptide chains are being constructed here at the site of the rough ER because the DNA instructions have come out. The ribosomes are reading that information and spitting their little proteins, their initial proteins, into the rough ER. The rough ER continues to move down the chain until it enters the smooth ER, where lipids and other things kind of get added on to the assembly of all of that stuff. They get kind of spun off into these little vesicles that take them to the Golgi body, where they are essentially finished up and they are basically made ready for transport. And then the vesicles actually pinch off from the Golgi bodies and transport all the way to the plasma membrane, where it merges with the plasma membrane and product gets released. So that's kind of what's going on, the order of events. We're going to learn the details of what happens right here at the rough ER, again, during our protein synthesis lecture. And this is just another view of how that happens. Here's the rough ER, and the DNA information comes out of the nucleus in the form of like a photocopy. The rough ER has all of the ribosomes on the surface, which read those photocopies and spit their little protein starter kits into the rough ER, which then moves into the smooth ER, where lipids and other additive things get kind of glommed on. And then you have a pinching off here and a vesicle that carries those products to the Golgi bodies, the Golgi apparatus, which then finishes it off and gets it ready for transport out of the cell. And here it is passing out of the plasma membrane. Happy days. How about that? Now, lysosomes are another kind of organelle which are kind of related to the endomembrane system, because you can actually use these little endomembrane pockets to take digestive enzymes and merge with products coming in to the cell. And you can actually use this to digest things. Or in some cases, you can digest old organelles. Like, let's say here's a mitochondrion. We're going to talk about them in just a second. Let's say you have a mitochondrion that's all worn out. Well, what you got to do is merge them with the lysosome. The lysosome actually releases a whole bunch of digestive enzymes, and it breaks down the mitochondrion and all of those molecules and stuff that break down as a result of it can then get recycled and reused for other purposes. These two processes here are known as phagocytosis. Phagocytosis means to actually eat or cell eating. So you're actually bringing in a food vacuole and then digesting it. And then this is called autophagy. Autophagy refers to eating oneself. In this case, an old, decrepit, and dilapidated organelle. You're breaking it back down so that you can recycle it. Phagocytosis, cell eating. Autophagy, self-eating, essentially. Okay, so this kind of summarizes that. Lysosome is that membranous sac that floats around in cytoplasm that does these things. They contain digestive enzymes. Now, all cellular activity requires energy. No matter what you're doing in the cell, you need energy to do that. So what is that source of energy? Well, we talked about those in the last few lectures in the biomolecules. Carbohydrates have lots of energy. Protein actually possesses some energy. And of course, fats, lipids contain lots and lots of energy. Of course, if you're a plant, your energy source is going to be sunlight, right? So these are sources of energy. And the structures that actually store up and use this energy would be the mitochondrion. Mitochondria are present in all animal and plant cells, and also fungi and protists. But remember, we're just talking about animals and plants here. All animals and plant cells contain mitochondria, but only plant cells contain chloroplasts. Okay, so let's focus first on the mitochondrion. Mitochondrion is singular, plural is mitochondria, and these are like the powerhouses of the cell. Energy gets produced in these in a process called ATP synthesis. This is the topic here of respiration and metabolism. Another lecture in the future here. Actually, that's what we're going to deal with next week. And it is here that you release huge quantities of energy so that the cell can actually do work. You could think of them like this, like power plants, like little energy factories. That's what they do. Okay, how do we actually acquire energy from this? Well, this is an oversimplified model. This is called aerobic respiration. This is what you do when you're breathing every single day. You ever wonder what it is that's going on when you're breathing? Well, you need oxygen, right? And you also need sugar. You probably knew that. You need sugar, you need oxygen. But what's going on? Because when you breathe out, you're releasing carbon dioxide, and it turns out also water vapor. You know that when you breathe on a mirror, you see water vapor released. And that spits out huge quantities of energy. And so this reaction actually requires oxygen. You cannot do this without oxygen. It actually works very differently in the absence of oxygen. You need oxygen. So if oxygen is necessary for this process, can you guess what kinds of cells are going to require more oxygen? Where are you doing the most work in your body? Okay, if you guessed muscle cells, then you're right. Not only muscle cells, though. Anything that has high metabolic activity, these are cells that are going to require lots and lots of mitochondrion because they're going to be having to produce lots and lots of energy all at one time. Now, there's something very interesting about mitochondrion. This is kind of a diagram of mitochondrion. A lot of the other organelles, you think about like the endoplasmic reticulum, which we just talked about, the Golgi bodies, and even the lysosomes, these are all kind of membranous sacs that possess just a single membrane. But look here at the mitochondrion. These guys are double membraned. Now, that's kind of odd. And actually, it puzzled scientists for a very long time until about the early 1990s, when we suspected that actually mitochondria might be very closely related to other prokaryotes. Well, that's kind of weird. It means that they're actually related to bacteria. It's like their most recent ancestor is an ancient bacteria. Now, let's take a look at the anatomy of the mitochondria here. The cristae are these infoldings of this inner layer here. And this is really important because this is actually the site of respiration, of metabolism that happens in your body to produce lots of ATP. It happens right here along this fold. And also, you'll notice the matrix. This is a compartment inside the membrane. And this barrier here, the site of this barrier between the matrix and actually this outside area is where the ATP synthesis occurs. It's across this barrier that happens. Again, this is the process of respiration that we're going to talk about a little bit later. Okay, let's contrast this double membraned organelle with this organelle right here. This is a chloroplast. Chloroplasts are really amazing little organelles inside of plant cells. Remember, they're not present in animals. It'd be really cool if they were, but they're not. They're only found in plants. This is the site of photosynthesis. And you'll also notice that chloroplasts, along with mitochondria, are actually double membraned as well. Kind of weird. They have an inner and an outer membrane. Inside here, inside this fluid matrix is called the stroma, and that's the inner fluid cavity, and this is where all of the sugars get produced in the process of photosynthesis. Yes, that's right, sugar is the actual product of photosynthesis. Everybody says, oh hey, plants are making oxygen for me. No, they're not. The oxygen is actually the waste product. The product that they're trying to make is actually the sugar. Okay, these little sacs here are the thylakoid membranes, or the thylakoids, if you will. This is where the green pigments, the actual chlorophyll pigments, are concentrated. So it is the site of the actual light reaction that allows photosynthesis to proceed. Okay, let's come back to both of these organelles now. Our mitochondrion and our chloroplasts. This is a really interesting new discovery. You see, there's a couple of other interesting things about chloroplasts and mitochondrion that are unlike any other organelle. They actually have their own DNA. That's really weird. You know, every cell has its own DNA, but here you have organelles that possess their own DNA separate from the actual DNA in the nucleus of the cell that they're living in. And the other thing is that their DNA is circular. Now, we haven't talked about the implications of this, but the only organisms on the planet that have circular DNA are bacteria. So there's many, many, many lines of evidence that actually point to these organisms being distantly related to free-living bacteria. Okay, what does this mean? How could this possibly be? Well, it is now pretty much set that the way we think about this is that at some point in our distant past, when eukaryotic cells were evolving, because these things resemble bacteria, at some point we probably acquired a particular bacterium, and instead of digesting that bacterium, we acquired it and actually retained whatever metabolic function the bacteria had. In this case, a bacterium, which eventually led to our modern mitochondria, was actually able to produce its own powerhouse, its own power source. So this is known as the endosymbiotic theory, the theory that bacteria were engulfed but not digested, and we have to this day retained the function of those bacteria. That's pretty darn amazing. Okay, now I do want to mention one particular plant organelle specialty, and that is the central vacuole. You do not find central vacuoles in animal cells. But you find them exclusively in plant cells and also in a couple of protist, plant-like protists. But these are really important to plants because they actually help add to the rigidity of this. So lots of central vacuoles in carrots, for example. They help maintain the rigidity of carrots. Here's another view of a central vacuole. It looks kind of like this. You can store all kinds of stuff in central vacuoles. As I had mentioned before, you can put sugars in there, you can put amino acids, you can store your waste products in there until you're ready to get rid of it. And when water flushes into the cell, it can actually retain it in there and increase the pressure inside the cell, and it presses out against the cell wall and gives that cell, and hence the plant that the cell is part of, a greater rigidity. Okay, you guys remember the lab number 2 in Biology 109 lab? If you haven't taken the lab, this is something you'll get ready to, or you can look forward to. This is what a typical plant cell would look like. You have this cell wall right here, and in the very middle you have this humongous vacuole, this huge cellular vacuole in the middle. And it's loaded with water in here and other stuff, but we'll just say water for now. Here's the cytoplasm, the stuff in blue. And all of the green, these are all of the chloroplasts that are kind of floating about in the cellular matrix right here. Well, if you were to add salt to this system, you would be putting these cells in a solution called a hypertonic solution, where there's a greater concentration of salt outside and hence a lower concentration of water outside. Well, what ends up happening is the water that is inside the cell actually rushes out as a result of this. The pressure actually causes the water to rush out, and the entire cell, the cell membrane, actually pulls away from the cell wall and shrinks the entire contents of the cell. This is called plasmolysis. So, something really interesting to look forward to when you eventually do take Biology 109 lab. Unless you've already taken it, then you know exactly what I'm talking about. Now, using this process, certain cells can actually move, and they do it by a process called cytoplasmic streaming. That is, they are actually able to flex certain parts of, or certain sides of the cell by pulling on these little microtubules, this little cytoskeleton, microtubule skeleton, and they force certain sides of the cell out and cause other sides to relax. And in so doing, an organism like this amoeba is actually able to, quote-unquote, walk across the bottom of a pond. And in fact, when it encounters a food particle like you see here, they're able to do this cytoplasmic streaming to engulf a food item and eventually eat it. That would be a really frightening thing, to be a little microbe in there and be eaten by an amoeba that is outrunning you. That's kind of interesting. Now, let's go to the cytoskeleton. We had mentioned this before. Remember, it is only present in eukaryotes, and it is this whole series of girders, joists, and support beams that are made of proteins that actually help retain the shape of the cell. Very useful, very important. It also helps anchor organelles. Now, if you think about your own body, if you were running in one direction, you would expect all of your organelles to go flinging to the back of each of the cells, right? Well, having a nice cytoskeleton in there prevents that from happening. It keeps everything kind of anchored in place, and it keeps it from moving around too much. Now, there are 3 main types of cytoskeleton components, and we're only going to cover them in a cursory fashion. We're not going to go into great detail on this. But the first one are called the microtubules. And even though the word micro actually sounds like small, microtubules are actually the largest of these 3 elements. They're the thickest. You could think of them like the huge iron I-beams in a building. They are the big, huge I-beams. They are the ones that kind of add the most structural support. Then there's the microfilaments. These are the thinnest ones. These would be more like the walls and that sort of thing, that add structural support just to a room per se. And in between all of this would be the intermediate filaments, which we're not going to really distinguish between microfilaments and microtubules. These are just things that actually connect these two systems together. And so microtubules, the longest elements, these have a huge array of functions. Not only do they maintain the structure inside the cell, but they actually can press out through the cellular membrane to create structures called cilia or flagella, which actually allow a cell to move. You can beat them back and forth, and it's kind of like having a tail. They can actually swim. So microtubules contribute to the shape of the cell. They also allow that cell to move in certain cases, and they become very important when you pull against them in cell division. They help keep everything in an orderly movement when you're dividing a cell. And they also guide organelle movement. So if you think of it, they actually create a system of roads and freeways. And if you were moving the proteins into and out of a cell, or whatever product it is you're making, you would use these girders. You would use these microtubules as kind of like a road to get out of the cell. You know that once you latch onto one, it's really easy to find the out, right? Okay, so cilia and flagella are the moving structures that I mentioned, and these are made up of microtubules as well. I'm going to show you an example in the next slide, but first I want to, I guess I can show it to you here. Cilia and flagella. This is kind of what they look like in cross section. They're made up of a series of proteins arranged in a spoke almost. And you can kind of imagine a big long tube. Here's just another variety of this. A big long tube made up of little mini protein tubes like this. If you were to tug on one side but not on the other, it would actually cause this thing to flex to one side. Okay? And then if you were to relax it and then tug on the other side, it would beat in the other direction. And in so doing, if you did this in synchrony, you could actually move. Now, at the very base of this is a structure called the basal body. Here's the basal body right there. And the basal body actually controls that flexing. I'm going to mention a motor protein here, but you're not going to have to know this in very great detail. One might ask, well, what is the actual motor mechanism that drives this flagella back and forth and causes the cell to actually move or have movement? Well, this is it right here. It's called the dynein. This is a very new discovery, everybody. This is less than a decade old to make it into our textbooks. And it actually drives the actual bending motion of these cilia. I urge you all to check out some of the videos that I posted on Blackboard, because I actually show some of these really cool animals, these little organisms, in fact, like little ciliates that swim around with this synchronic movement where they're beating their cilia back and forth. Here's another one that a lot of people are familiar with, sperm. Sperm are single-celled organisms that move with this dynein beating metasynchronic movement of a tail. And they're able to swim that way. This is with a single flagellum, but if you have thousands of them and they're small, we know them as the cilia, like this guy right here, which is ciliated. It looks furry, but in fact it's got thousands of little small microtubules projecting out from the cell membrane, and they beat them together to form movement. That's pretty amazing. Now, where else do we have this? Well, how about like every cell in your lungs? All your lung cells on the surface of your lungs, on the inside, possess lots and lots of cilia. And by having these cilia kind of beat at the same time, they're able to move mucus and other noxious, cruddy stuff out of your lungs. We also have this present in our small intestines. It helps keep the food moving in the right direction. Think about how important that is, right? What if the food wasn't moving in the right direction? You'd be dead. So you really do rely on these microtubules, these microtubule projections. Now, why is it bad for smokers? Why is it that smokers hack all the time and have to cough to get rid of their phlegm? Well, it's because they've successfully killed all of these tracheal cells and basically burned off the microtubule tips to the cilia, and it doesn't work anymore. So that's not a good thing. I urge you all to quit smoking if you're a smoker. Okay, now what other cell surface specializations can we come up with? There are a myriad of them. Go out into the ocean environment and pretty much every microbe you encounter is going to have some strange new cell surface feature to them. This is just one such example. These are little critters called radiolarians. Don't memorize that. You don't need to know that. But just realize that they are marine creatures that have a really weird surface that serves their purpose. It's actually a shell. Here is a single-celled organism that produces a shell. Now, let's talk about cells in plants in particular. Cell walls. We talked about cell walls being really important, right? These are semi-rigid structures that enclose the entire cell membrane. Yes, plant cells do have a cell membrane, but you don't normally see it because it's lying flush up against the cell wall. Um, it helps it maintain its shape. It resists rupture. You can punch a carrot and it still keeps its shape. That's kind of nice. And so, who has a cell wall? We talked about all plants having them, but it turns out fungi also possess cell walls, and so do some protists. Not very many, but some do. And plant cell walls are actually more complex than I'm making them out to be. They're actually made up of a series of layers that are very rich in carbohydrates and a few other things, sometimes proteins, but most often carbohydrates. And they actually butt up against each other and are glued together with some of these cell-to-cell junctions, which I'm going to show in just a second. We know them as plasmodesma. Plasmodesma actually connect the cytoplasm from one cell to another by creating a little conduit between the plant cell walls. That's kind of interesting. Plant cells can actually communicate with each other. How cool is that? And you just thought they were dumb things that sat there in the ground. Now, animal cells also have hard stuff around them sometimes. Think about some animal cells in your body. How about like skeletal elements, skeletal cells? Yeah, obviously those are producing hard parts too, but those are cells that are producing calcium carbonate. Very similar to plant cells producing carbohydrate-rich walls. So sometimes they're surrounded by a matrix that is secreted on the outside of the cell, producing this hard, bony material. And cartilage is made the exact same way. Cartilage and bone both made by animal cells that basically secrete this. Now, just as in plant cells are communicating with each other through these plasmodesma. Animal cells are doing the exact same thing. Of course, we don't call them plasmodesma in animal cells. We would call them adhesion junctions, or tight junctions, or gap junctions. Either way, these are essentially conduits or little tunnels from one cell to another. Think about like animal cells, skeleton cells. Skeletal cells in animal cells are surrounded by hard stuff, but they have passageways between the cells that actually allow them to communicate. Okay. So this is the summary now of all of the major organelle types that I want you to know for the midterm coming up in week number 5. And this is the summary of the cell. And the question you must ask yourself is, can I identify, define, and give the function of every single one of these organelles? That's right, you're required to know all of them. The only one I don't have circled here is the centriole and also the nuclear pores, although you should know the nuclear pores as the little holes through which the DNA material, the photocopied DNA material, gets delivered out through. But the centrioles, we're going to talk about them later when we get to cell division, mitosis. But otherwise, everything else here, I would get a piece of paper out, write them all down, and be able to define them. That would be a really good thing.